Welcome back to the deep dive. Today we're jumping right into the instruction manual for life itself. And maybe the best way to start is with this picture. Imagine you're on this sunny Italian island, Asinara, and you see these wild albino donkeys.
Ah, yes, the Asinara donkeys.
Yeah, stark white fur, these really striking pink eyes. It's such a contrast to the usual brown donkeys.
It really is. And what's incredible is that whole difference, that specific look, it all comes down to a single faulty gene.
One gene.
One gene. A tiny mistake in the instructions for making a pigment protein, or rather the enzyme that helps make pigment.
Wow, it's such a visual, right? Because this isn't just about donkeys, is it?
Not at all. It's a perfect visible example of a fundamental process. This process governs basically all inherited traits. Your hair color, how your cells work,
Everything.
right down to the enzymes.
Exactly. In those donkeys, that faulty gene means they can't make a working version of an enzyme called tyrosinase. It's essential for making pigment. So, no pigment. It's a really clear line from gene to trait. So that's what we're tackling in our deep dive today. Gene expression, from gene to protein. We want to take you on this amazing molecular journey. How does the info stored in your DNA, the blueprint, actually get used?
How does it get read and turned into the proteins that make you, well, you?
Exactly. We'll look at how genes determine traits, the key steps, transcription and translation, how RNA gets processed, and how tiny changes, mutations, can sometimes have huge consequences.
It's your shortcut to understanding how lice instructions are read and acted upon.
And underpinning all of this is a core concept, right? The sort of flow of information.
That's right. It's what Francis Crick famously called the central dogma of molecular biology. It's this directional flow. DNA holds the master instructions. Those instructions are copied into RNA.
Okay.
And then RNA is used to build proteins. DNA to RNA to protein. That's the fundamental flow in, well, pretty much all life we know.
DNA to RNA to proteins sounds simple when you put it like that, but figuring it out must have been quite the journey. Where did that understanding even start?
Well, the very first hints actually came quite early. We have to go back to 1902 to a British physician named Archibald Garrod.
1902.
Yeah, he was really ahead of his time. Garrod suggested that genes influence traits by controlling the production of enzymes, the proteins that speed up chemical reactions in cells.
How did he connect genes and enzymes back then?
He studied inherited diseases, like one called alkaptonuria, where people have very dark urine. Black, almost. He figured out it was because they couldn't break down a specific chemical.
Okay.
And he reasoned, well, maybe they're missing the specific enzyme needed for that breakdown, so the chemical just builds up and gets excreted. He was linking heredity, chemistry, and disease in a completely new way.
So he laid the groundwork, but the real proof came later.
It did. Fast forward to the 1930s and 40s. George Beadle and Edward Tatum did some absolutely groundbreaking work, using, believe it or not, common bread mold.
Bread mold.
Wow.
Neurospora crasta, right?
That's the one. Their experimental setup was really clever. They used X-rays to cause mutations in the mold, basically breaking specific genes.
Okay, creating mutant mold.
Right. Then they checked if these mutants could still grow on a very basic, minimal nutrient medium. If a mutant couldn't grow unless you added, say, a specific amino acid like arginine,
Yeah.
it meant the gene needed to make the enzyme for synthesizing arginine must have been damaged by the X-rays.
Ah, I see. So if it can't make arginine, it needs it supplied, linking a specific gene defect to a specific missing enzyme.
Exactly. This led them to their famous one gene, one enzyme hypothesis. It was a huge step, earned them a Nobel Prize, fundamentally changed biology.
One gene, one enzyme. Got it. But like always in science, it wasn't the whole story, was it?
No, it gets refined. People realize, well, not all proteins are enzymes. Think about keratin in your hair or insulin, the hormone. They're proteins, but not enzymes.
Right. Structural proteins, signaling proteins.
Precisely. Yeah. And also, some proteins are made of multiple different polypeptide chains, like hemoglobin, the oxygen carrier in our blood. It has 4 chains, and each type comes from a different gene.
So the hypothesis needed an update.
It evolved into the one gene, one polypeptide hypothesis, which is closer. But even that isn't the full picture today, thanks to something called alternative splicing, which we'll definitely get into.
Alternative splicing. Sounds intriguing. Okay, we'll park that for a moment. Let's talk about the molecules. DNA has the info, proteins do the work. What's the go-between?
That crucial intermediary, the bridge, is RNA, ribonucleic acid.
How's it different from DNA?
Chemically very similar, but a few key differences. It uses a different sugar ribose instead of deoxyribose. It uses the base uracil, U, instead of thymine, T. And RNA is usually single-stranded, unlike DNA's double helix.
And it acts as the messenger.
Exactly. It carries the instructions from the DNA, which stays safe in the nucleus in eukaryotes, out to the protein-building machinery.
Which brings us to the two main stages, transcription and translation. Let's break those down. If DNA is the master recipe book.
Then transcription is like carefully copying one specific recipe from that book onto a portable index card. You're rewriting the information from DNA language into RNA language, specifically messenger RNA or mRNA.
Copying the recipe. Same language, just a different format.
Pretty much. The sequence of DNA bases dictates the sequence of RNA bases. A becomes U, G becomes C, and so on.
Okay, index card in hand. Now what?
Now comes translation. This is where the language really changes. You take that mRNA index card and use its instructions to actually build the dish, the protein.
So translating from the nucleotide language of mRNA into the amino acid language of proteins.
Precisely. The sequence of codons on the mRNA tells the cell which amino acids to link together in what order. And this whole process happens on complex molecular machines called ribosomes.
Ribosomes, got it. And where these things happen in the cell, that differs between simple and complex life.
It does, and it's an important difference. In bacteria prokaryotes, they don't have a nucleus. So transcription and translation can happen basically at the same time in the same place, the cytoplasm.
Like an open workshop?
Kinda, yeah. But in eukaryotes like us, plants, fungi, we have a nucleus. The DNA is kept inside. So transcription happens in the nucleus. The resulting RNA molecule, called pre-mRNA at this stage, then gets processed, modified.
Ah, the processing step.
Right, and then the finished mRNA travels out of the nucleus into the cytoplasm, where translation happens on the ribosomes. That separation allows for extra layers of control in that crucial RNA processing.
Okay, let's zoom in on that mRNA message. How is the code written? You said codons.
Right, the genetic code is read in triplets. 3 nucleotide bases form a word, or a codon. Think about it. You've only got 4 RNA bases, A, U, G, C.
Yeah. Okay.
If codons were only 2 bases long, you'd only have 4 times 4, 16 possible combinations. But there are 20 different amino acids used to build proteins, not enough codes.
Ah, so you need 3.
Exactly. 3 bases gives you 4 times 4 times 4, which is 64 possible codons. It's more than enough to code for all 20 amino acids, plus some stop signals to end the protein chain.
64 codes for just 20 amino acids and stop signals. Seems like overkill.
Well, it means the code is redundant or degenerate. Most amino acids are specified by more than one codon, like GAA and GAG both code for glutamic acid.
Does that redundancy matter?
Oh, definitely. It provides a buffer against mutations. A change in the third base of a codon might not even change the amino acid, but importantly, the code is not ambiguous, meaning that any single codon specifies only one amino acid or stop. GAA always means glutamic acid, never anything else. No confusion there.
Okay, triplets. And reading them correctly is the vital, right? The reading frame.
Yeah, absolutely critical. The ribosome reads the mRNA codons sequentially, 3 bases at a time without overlapping.
Okay.
If you shift that starting point by 1 or 2 bases, disaster. Total gibberish. Imagine the sentence. The fat cat ate the rat. If you start one letter off, you get H if ATC. 88TT or 8T. Makes no sense. A frame shift mutation usually leads to a completely non-functional protein.
How did scientists even crack this code? It seems so complex.
It took some brilliant work in the early 1960s. Marshall Nirenberg was key. He made artificial mRNA molecules consisting of just one repeating base, like poly-U, just UUU.
Okay.
He added this poly-U mRNA to a test tube system that could make proteins. And what he got out was a polypeptide chain made only of the amino acid phenylalanine.
So UU must code for phenylalanine.
Bingo. By using different artificial mRNAs, they systematically figured out the amino acid for each codon. All 64 codons were deciphered by the mid-60s, a monumental achievement.
And this code, it's the same everywhere, in bacteria, in us.
It's nearly universal. The genetic code used by a bacterium is essentially the same as the one used by a human, or an oak tree, or a mushroom. There are a few very minor exceptions in some organisms or organelles, but the basic code is conserved across almost all life.
That's profound. What does that tell us?
It's incredibly powerful evidence for a common ancestor for all life on Earth. We all inherited this fundamental operating system. It's why genetic engineering works. You can put a human gene into bacteria, and the bacteria will read the codons the same way and make the human protein, like insulin.
Amazing. Okay, let's dig into transcription itself. Copying DNA to RNA. Who's the main player?
The key enzyme is RNA polymerase. It's the machine that builds the RNA strand. It pries apart the DNA double helix temporarily and uses one strand as a template.
Okay.
It then adds complementary RNA nucleotides, matching A with U and G with C, linking them together to form the growing RNA chain. And unlike DNA polymerase, RNA polymerase can start a chain from scratch. It doesn't need a primer.
How does it know where to start and stop on the DNA?
There are specific DNA sequences that act as signals. The start signal is the sequence called the promoter. RNA polymerase binds here.
The promoter.
Right. And in bacteria, there's usually a stop signal sequence called the terminator. The stretch of DNA between the promoter and terminator that gets copied into RNA is called a transcription unit.
So let's walk through the steps. Initiation first. Getting started.
Okay, initiation. RNA polymerase has to find and bind to that promoter sequence. In bacteria, it's relatively straightforward, but in eukaryotes, it's more complex.
How so?
Eukaryotes need several helper proteins called transcription factors. These proteins have to bind to the promoter region first, often to a specific sequence within it called the Tata box.
Tata box. Okay.
Once these factors are in place, then RNA polymerase II, the main one for making mRNA, can bind. This whole assembly of factors and polymerase at the promoter is called the transcription initiation complex.
A whole team effort just to get started.
Exactly. It allows for much finer control over which genes get turned on when. Once the complex is formed, the DNA unwinds, and the polymerase is ready to go.
Then comes elongation, building the RNA chain.
Right. The polymerase moves along the DNA template strand, reading the DNA bases and adding the corresponding RNA nucleotides to the 3' end of the growing RNA molecule.
Synthesizing in the 5' to 3' direction, like DNA replication.
Correct. As it moves along, the DNA double helix unwinds ahead of it and rewinds behind it. And this can happen multiple times on the same gene simultaneously.
Like a convoy.
Yeah, you can have multiple RNA polymerase molecules following each other down the gene, each making an RNA copy. It allows the cell to make a lot of RNA and thus a lot of protein relatively quickly.
Efficient. And how does it stop? Termination.
The mechanisms differ a bit. In bacteria, the polymerase transcribes that terminator sequence we mentioned. This signal causes the polymerase to detach from the DNA and release the finished RNA transcript. It's ready to be translated immediately.
But eukaryotes are different again.
They are. Eukaryotic RNA polymerase second actually transcribes past the sequence that will eventually be the end of the mRNA. It transcribes a specific sequence called the polyadenylation signal, AUAAA in the RNA.
AAUAAA.
Shortly after this signal appears in the growing RNA strand, proteins associated with the transcript recognize it and cut the RNA free from the polymerase, maybe 1035 nucleotides downstream. The polymerase eventually falls off the DNA too.
And this RNA that's just been cut free, that's the pre-mRNA you mentioned, still needs work.
Exactly. In eukaryotes, this primary transcript, this pre-mRNA, has to undergo significant processing before it can leave the nucleus and be translated.
Okay, RNA processing in eukaryotes, what happens? You mentioned caps and tails.
Right. Two key modifications happen at the ends. The 5' end, the first end made, gets a special modified guanine nucleotide added. This is the 5' cap.
Oh, a G cap.
Yep. And the 3' end, where it was cut free, gets a long chain of adenine nucleotides added, the poly A tail. Can be 50 to 250 as long.
Why add these? What did the cap and tail do?
They're crucial. They seem to help with exporting the finished mRNA out of the nucleus. They protect the mRNA from being degraded by enzymes in the cytoplasm. And they help the ribosomes bind correctly to the 5' end to start translation, essential functions.
Okay, caps and tails. What else happens during processing? This is where splicing comes in.
This is the really amazing part. RNA splicing. See, most eukaryotic genes and their RNA transcripts have long stretches of non-coding sequences called introns, kind of like interruptions.
Introns, interspersed with the coding bits.
Exactly. The coding regions are called exons. They're the parts that are eventually expressed. Splicing is the process of cutting out all the introns and joining the exons together precisely.
Like editing out the commercials from a recorded TV show?
That's a great analogy. You need to remove the non-essential parts, introns, and stitch the important segments, exons, back together seamlessly to get the final message.
How's this editing done? It sounds like it needs to be incredibly precise.
It is. It's carried out by a large, complex molecular machine called the spliceosome. Spliceosomes are made of proteins and small RNA molecules. These small RNAs actually help recognize the splice sites, the boundaries between introns and exons, and might even catalyze the splicing reaction itself.
RNA doing catalysis, like an enzyme.
Precisely. This leads us to the concept of ribozymes RNA molecules that function as enzymes. The discovery that RNA could be catalytic was revolutionary. It broke the old all enzymes are proteins rule. RNA in ribosomes is another key example.
Wow. Okay, so splicing removes introns. But you mentioned alternative splicing earlier.
Ah, yes. This is where it gets really powerful. Because genes have multiple exons, a cell can sometimes splice the RNA transcript in different ways. It might treat a particular stretch as an exon in one context, but as part of an intron to be removed in another context.
So from the same gene, the same pre-mRNA.
You can generate different mature mRNA molecules, which are then translated into different, though related, proteins.
That's incredible. So that explains how we can have, what, maybe 20,000 genes, but make way more proteins?
Exactly. Alternative splicing vastly increases the protein coding potential of our genome. It's a major reason for the complexity of vertebrates, including us. One gene can lead to multiple proteins with different functions. It's a huge layer of regulation and diversity.
Mind blown. Okay. So after capping, tailing, and splicing, we finally have mature mRNA, ready for translation.
Ready to leave the nucleus and find a ribosome in the cytoplasm.
Let's talk translation then. RNA to protein. We need an interpreter molecule, right?
We do. That's the job of transfer RNA, or tRNA. These are relatively small RNA molecules, about 80 nucleotides long.
And they have a specific shape.
Yeah, they fold up into a specific 3D structure, kind of an L shape, though often drawn flat like a clover leaf. Crucially, they have two important ends.
Okay.
At one end, there's an attachment site for a specific amino acid. At the other end, there's a loop with 3 nucleotides called the anticodon.
Anticodon, that matches the codon on the mRNA.
Precisely. The anticodon on the tRNA base pairs with the complementary codon on the mRNA strand within the ribosome. This is how the tRNA ensures that it delivers the correct amino acid corresponding to that specific mRNA codon.
So each tRNA is specific for one amino acid and recognizes the right codon for it. How does the tRNA get loaded with the correct amino acid in the first place? That seems critical.
Absolutely vital for accuracy. This is done by a family of enzymes called aminoacyl tRNA synthetases.
That's a mouthful.
It is. But their job is crucial. There are 20 different synthetases, one for each of the 20 amino acids. Each synthetase recognizes one specific amino acid, A and D, all the tRNAs that should carry that amino acid. Remember, the code is redundant. It then uses energy from ATP to covalently attach the correct amino acid to its specific tRNA. This is often called charging the tRNA. It's a critical quality control step.
Ensuring the right amino acid gets linked to the right interpreter molecule.
Got it.
Yeah.
And what about this wobble idea? Does that affect tRNA pairing?
It does. The base pairing between the third nucleotide position of an mRNA codon and the corresponding base of the tRNA antagodon is a bit more flexible. It can wobble.
Meaning a single tRNA anticodon might be able to recognize more than one codon as long as the first two bases pair perfectly. For example, a tRNA with the anticodon UCU could potentially pair with mRNA codons, AGA or AGG, both of which code for arginine. It adds flexibility and explains why we don't need 61 different tRNAs for the 61 amino acid codons.
Clever. Okay, so we have mRNA, charged tRNAs. Where does the actual protein building happen? The ribosome?
The ribosome, these are complex structures made of ribosomal RNA, RNA and proteins. They have two subunits, a large one and a small one, that come together on the mRNA.
And they manage the whole process.
They coordinate everything. They provide the sites for mRNA binding and for the tRNAs to come in and match their anticodons to the mRNA codons. They also catalyze the formation of the peptide bonds that link the amino acids together.
rRNA doing catalysis again.
Yes. The ribosome is essentially a giant ribozyme. The rRNA, not the protein, is the main catalyst for peptide bond formation.
Okay, what are the key sites on the ribosome?
There are 3 main binding sites for tRNA, usually called the A, P, and E sites. The A site, aminoacyl tRNA site, is where the next incoming charged tRNA carrying its amino acid binds.
A for arrival, maybe.
Good way to think of it. The P site, peptidyl tRNA site, holds the tRNA carrying the growing polypeptide chain. P for polypeptide.
Okay.
And the E site, exit site, is where the empty tRNA, having delivered its amino acid, is discharged from the ribosome. E for exit.
A, P, E, arrival, polypeptide, exit. Makes sense. So let's build a protein. Initiation first.
Right. Initiation brings together the mRNA, the first tRNA carrying methionine, the universal start amino acid, and the 2 ribosomal subunits. The small subunit binds the mRNA, usually near the 5' cap in eukaryotes, and scans for the start codon, AUG.
AUG signals start here.
Correct. The initiator tRNA with methionine binds to the AUG. This is critical because it establishes the correct reading frame for the entire mRNA. Then the large ribosomal subunit binds form the complete translation initiation complex. The initiator tRNA sits in the P site.
Okay, complex formed, reading frame set, now elongation. Adding more amino acids.
Elongation proceeds in a three-step cycle. First, codon recognition. The next mRNA codon in the A site attracts the complementary tRNA carrying its amino acid.
tRNA arrives at the A site.
Second, peptide bond formation. The ribosome, specifically rRNA in the large subunit, catalyzes a peptide bond between the amino acid on the A site tRNA and the growing polypeptide chain attached to the P site tRNA. The polypeptide chain is transferred from the P site tRNA to the A site tRNA.
The chain grows.
Third, translocation.
Right.
The ribosome moves one codon down the mRNA. The tRNA that was in the A site, now carrying the polypeptide, moves to the P site. The empty tRNA that was in the P site moves to the E site and is released. The A site is now empty, ready for the next charged tRNA.
And the cycle just repeats. Codon recognition, peptide bond, translocation.
Over and over, adding amino acids one by one, elongating the polypeptide chain from its N-terminus methionine end to its C-terminus carboxyl end.
Until, how does it stop?
Until a stop codon, UAG, UAA, or UGA, on the mRNA enters the A site. There are no tRNAs with anticodons for stop codons.
So what binds?
A protein called a release factor binds to the stop codon in the A site. This binding triggers the hydrolysis, the breaking of the bond between the completed polypeptide and the tRNA in the P site.
Protein release.
Polypeptide released. And then the whole complex ribosome subunit's mRNA release factor falls apart, ready to start again on another mRNA if needed.
Is that released polypeptide chain immediately a working protein?
Often not quite. First, it has to fold up into its correct, unique three-dimensional shape. That's largely dictated by the amino acid sequence itself. But then many proteins need post-translational modifications.
Like what?
Oh, things like adding sugars or lipids, maybe removing the initial methionine, sometimes cutting the polypeptide into smaller active pieces, or joining together multiple polypeptide chains, like in hemoglobin. Lots of possibilities to make it fully functional.
And getting the protein to the right place in the cell? You mentioned targeting earlier.
Right. If a protein is destined for secretion, or insertion into a membrane, or specific organelles like the ER or Golgi, its synthesis usually starts on free ribosomes, but then gets directed to the ER membrane.
How does that happen?
The polypeptide starts with a signal peptide, a short sequence at the N-terminus. This acts like an address label. A complex called the signal recognition particle, SRP, binds to this signal peptide, temporarily halts translation, and escorts the whole ribosome complex to a receptor protein on the ER membrane.
Docking at the ER.
Exactly. Translation then resumes, and the growing polypeptide threads through a channel into the ER lumen or gets embedded in the ER membrane. Proteins destined for other places like mitochondria or chloroplasts have different targeting signals.
It's like a cellular postal system. Amazing coordination. And cells can make lots of one protein quickly.
Yes, using polyribosomes or polysomes. A single mRNA molecule can be simultaneously translated by many ribosomes trailing along it, like beads on a string. Mass production.
And in bacteria, that coupling you mentioned.
Right, in bacteria, because there's no nucleus, translation can begin on an mRNA molecule, even while transcription of that same mRNA molecule is still in progress. It's incredibly efficient for them.
Whereas in eukaryotes, the separation allows for all that RNA processing.
Correct. The nuclear envelope allows time and space for capping, splicing, tailing, complexity, and control.
Okay, so that's the journey from gene to protein. But what if there's a typo in the original DNA blueprint? A mutation?
Mutations, changes in the genetic material, are fundamental. They are the ultimate source of new genes, new alleles, the raw material for evolution. Life wouldn't change or adapt without them.
We often think of them as bad, but they're essential for diversity.
Absolutely. Now, many mutations are small scale, affecting just one or a few nucleotide pairs. These are called point mutations.
And these can cause diseases.
They certainly can. Sickle cell disease is a classic example. A single nucleotide change in the gene for hemoglobin changes one amino acid, causing the protein to misfold and red blood cells to sickle. Familial cardiomyopathy, a heart condition, is another example linked to point mutations in muscle protein genes.
What types of point mutations are there? Substitutions?
Yes, nucleotide pair substitutions are common, and they can have different effects. A silent mutation is when the nucleotide change still codes for the same amino acid, thanks to that redundancy in the code. No effect on the protein.
Okay, lucky break.
Then there's a missense mutation. Here, the change results in a different amino acid being put in. The effect can range from basically nothing, if the amino acid is similar or not in a critical spot, to completely devastating if it alters the protein's function, like in sickle cell.
Depends on the change and where it happens.
Exactly. And the third type is a nonsense mutation. This is when the change converts a codon for an amino acid into a stop codon.
UAG, UAA, or UGA.
Right. This causes translation to stop prematurely, resulting in a shortened, truncated protein that's almost always non-functional.
Okay, silent missense nonsense. What about adding or deleting bases?
Insertions and deletions. These can be even more disruptive than substitutions, especially if the number of nucleotides inserted or deleted is not a multiple of 3,
Why not a multiple of 3?
because it causes a frameshift mutation. Remember the reading frame. Adding or losing 1 or 2 nucleotides shifts the entire triplet reading frame downstream for the mutation point.
Ah, like the fat cat example becoming HATC-ATA.
Exactly. All the codons downstream are wrong, leading to a completely different amino acid sequence, expensive missense, and usually hitting a premature stop codon fairly quickly. Frame shifts almost always result in a non-functional protein.
So insertions deletions are often worse than substitutions unless they're in multiples of 3. Where do mutations come from?
They can arise spontaneously, just as errors during DNA replication or recombination. Our cells have proofreading and repair systems that catch most of these, but some slip through. The error rate is incredibly low, maybe 1 in 10 billion nucleotides, but our genomes are huge, so errors happen.
And they can be induced.
Yes, by mutagens, physical or chemical agents that damage DNA and increase mutation rates. Physical mutagens include high-energy radiation like X-rays or UV light from the sun, which can cause specific DNA lesions.
Like thymine dimers.
Right. Chemical mutagens are diverse. Some mimic DNA bases and get incorporated incorrectly. Others insert themselves between bases, distorting the helix. Some directly modify bases, changing their pairing properties. And many cancer-causing chemicals, carcinogens, are mutagens highlighting the link between DNA damage and cancer.
Wow. So we've traced this incredible path from a single gene difference in those Acinera donkeys through transcription, RNA processing, translation, all the way to the protein and the consequences of errors.
It's an amazing, intricate system. DNA in the nucleus gets transcribed to RNA, that RNA gets processed, travels out, and is translated by ribosomes into proteins, which then fold and function, determining traits.
And our idea of what a gene even is has evolved quite a bit through all this discovery.
It really has.
Yeah.
From just an abstract concept of inheritance to a spot on a chromosome to a DNA sequence. Now, we really define a gene in functional terms. It's a region of DNA that can be expressed to produce a final functional product, which can be either a polypeptide or an RNA molecule.
Or an RNA molecule.
Right.
Because tRNAs and RNAs are crucial functional products coded by genes, but they aren't proteins.
Exactly. That broader definition captures the reality better.
So wrapping this up, maybe a thought for you, our listener. Think about the sheer complexity and the incredible fidelity of this whole process we've just walked through. Gene expression.
Millions of steps happening constantly.
Right. What does the precision needed tell you about the evolutionary pressures that shape these mechanisms? How do you balance that need for accuracy to maintain life?
With the necessity of allowing some errors, some mutations, which are the very source of the diversity and adaptation that drives evolution.
It's a fascinating balance, precision and change working together.
A truly remarkable system at the heart of life.
Definitely something to ponder. Thank you so much for joining us on this deep dive into gene expression. We hope you feel more equipped to understand this fundamental process. We'll catch you next time.